Um, Now, so there are different types of questions. So I'm just giving you a few examples here. It doesn't mean that these are the questions that will come out. I'm just give you an example. I submitted my question. I don't remember what I submitted. So it may come up similar. It may not come up exactly the same. But the important thing is you understand what it is, and then you'll be able to answer the questions. I personally think that among all the different disciplines, genetics, medical genetics is the easiest. If you ask your seniors, I'm not sure whether they'll tell you the same thing, but to me it's easy because you don't have to remember much. You only need to remember some very basic things. Then with the understanding, it is the understanding that is important. With the understanding, you'll be able to answer any type of questions. Okay? All right, so let's get first question. So there's only one answer here. It says a recessive mutant gene on chromosome 10, blah, 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 blah. Don't try to memorize anything. Don't try to recall from your lecture, but understand the question first. Let's see. Okay, so you see here it tells you it's a recessive mutant gene, and it tells you it's on chromosome 10. Does it matter whether it's on chromosome 10, chromosome 1, chromosome 2, chromosome X, chromosome Y or not? The fact that it tells you it's chromosome 10, meaning that you know chromosome 10 is in, is it too small? Can you see this autosome?
Yes.
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 Now, so you know that chromosome 10 is one of the autosomes, and it tells you it's a recessive mutant. So basically, you're looking at autosomal recessive, correct?
Yeah.
Now, so you have gone through autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. So now autosomal recessive. Now, so option A, this mutant gene on chromosome 10 of a female is inherited preferentially. See, it says it's preferentially. Did I click the right thing or not? No. Preferentially by the male offspring. Whenever you see the word autosomal, autosome is nothing to do with gender. So nothing to do with male or female. So this one is wrong. All right. Then it says affects both male and female equally. See, if it's autosome or autosomal recessive, both male and female offspring, they will be affected equally. They have equal chance to inherit the mutant allele. They also will be affected equally, no preference. So this one most likely is true, most likely, okay? Affects a heterozygous male. What is a heterozygous male? A heterozygous male, if it's autosomal recessive, a heterozygous male, what is the genotype? It would be capital A, small a, assuming capital A is the normal allele and small a is the mutant allele. So is this affected? Yes or no?
No.
No.
So how do you call this? For autosomal recessive, if this is the genotype, how do you call this guy? Carrier.
Carrier.
Yes, very good. You're right. Exactly, carrier. So it's a carrier. Same thing for option D. Heterozygous female is also capital A, small a, and she's also a carrier. Therefore, the answer is B. Okay? All right. So see, nothing much to remember. Oops. How do I scroll down? I have to go back to words, I think. All right. Let me clear the rubbish first. How come I hear music? Someone playing music? All right, let's go. Okay, question number 2. Mr. Tan, and the genetic disorder that he has is he is heterozygous for a autosomal dominant genetic disorder, right? The wife, Mrs. Tan, normal genotype. Okay, and the first child is a boy and also affected by the same disorder. So first, let's look at what is the genotype of Mr. Tan. Mr. Tan is heterozygous for an autosomal dominant. So his genotype would be capital A, small a, where the capital A is the mutant, is the dominant mutant allele, and the small a is the recessive allele, right? The normal allele. Mrs. Tan, she has normal genotype, so she would be first child boy affected by the same disorder, right? This is autosomal, and then this is so it doesn't matter whether it's a boy or a girl, but he is affected by the same disorder, so he is also carrying the mutant allele. Now, when we talk about autosomal dominant genetic disorder, most of the time it is heterozygous, right? This is heterozygous. Rarely it's going to be homozygous. There are various reasons. In order for an autosomal dominant, someone who is autosomal dominant, who has autosomal dominant genetic disorder to be homozygous, that is to have both big capital allele, meaning that both the parents, both the father and the mother must carry the mutant allele. That chance is not very high as compared to either the father or the mother has the mutant allele. Then he came up to a heterozygous. Okay. Another reason is most of the autosomal dominant genetic disorder, if they are homozygous, most likely and very likely that they don't survive. Okay. So that's the reason. So now it says, examine this situation. So you study the information given and then state your conclusion. So now look at A. All of his daughters are heterozygous. Sometimes you just read the statement like this, we can't really know whether it's correct or not correct. So I always emphasize that it's very important to use Punnett square. Now, I try to draw a Punnett square here. Oh, too bad. How come it doesn't draw? I thought I'm going to fail my art. It's so much easier to draw it on a whiteboard. Okay. All right. So his genotype is capital A, small a. The wife is, so if you draw the Punnett square, yes.
May I know why the wife is AA small? Why not the BAA?
Oh, here I didn't give you the how to say, the definition. For autosomal dominant, usually the capital A is the mutant allele. So maybe I should put you capital A. Better type it out. Capital A. Okay, so small a is the normal. If you refer to the notes that I given you, so capital A is the dominant mutant allele and small a is the normal allele. So she is too small a. So if you draw the Punnett square, did I answer your question? Hello? Who asked the question just now? Who asked the question just now? Did I answer your question?
Yes.
Okay, so if you're not clear, you have to tell me, anyone, if you ask question and I explain, if you're not clear, you have to tell me. So this is a Punnett square. Punnett square is very important. Always make use of it, because from here you will know that. Okay, so here you can see that 50% uh, among the offspring, they have 50% chance to be heterozygous, is affected, and 50% chance to be normal. So here it says all of his daughters are heterozygous. Well, this is an autosomal disorder. So the daughters could be either heterozygous or homozygous. So this one cannot be the answer. His son has 100% probability to be affected. Well, if you look at the Punnett square, no, he has 50% chance to be affected. It says that second child has 50% probability to be affected. So the first child is affected. Second boy, you go back to the same Punnett square, it will be the same probability. So second boy, right? What is the percentage of the second boy to be affected? 25%, 50%, 75, 100? Which one? Anybody? 50, right? You go back to the Punnett square. So this is 50% chance. So this one is the answer. How come it didn't? Right? So 50%. His son has higher probability of expressing the affected phenotype than the daughter. Now, always remember we're talking about autosomal dominant. Autosomal, so nothing to do with male or female. Okay? So this one is wrong. So the answer is C. You got it? Any question?
Yes.
Yes.
Got it.
So no question, huh? All right, let me
No.
clear this one. So let's move on to the next one. Okay. Mrs. Lin is heterozygous. She is heterozygous for an X-linked dominant genetic disorder while her husband is normal. Mrs. Lin is now pregnant, but the gender of the fetus is unknown. Calculate the probability of the offspring. Heterozygous. So what would be her genotype? X-linked dominant. So it will be an X. So X-linked dominant.
Capital E.
Small e. Correct? Because she's heterozygous, so capital L-U and capital A and small a. It's an X-linked dominant, so she carries one dominant mutant allele. She actually expressed the affected phenotype. So now it says the husband is normal. So what would be the husband's genotype? Husband is normal, so husband is X, capital A or small a. Remember here, capital A is the dominant mutant allele. So this is a small a. Don't put anything on the Y, please. Because this is talking about X-linked, right? I just finished marking the medical program first professional exam. When we asked about X-linked, many of them, they put something at the Y, and I'm so disappointed. Don't put anything at the Y if it is X-linked. Okay, so XAY. All right, so that is his genotype. So draw a Punnett square. Draw a Punnett square. Let's see. What's the better way to do? What is this? Is there somewhere I can put a table? Let's try again. So I'm drawing the Punnett square. This time I do better. Okay, so she is XBA and X small a. He is X small a y. Oh, my box too small. XBA and X small a. Oh, I'm getting better in drawing this. I hope my finger won't cramp after this session. Drawing with a mouse is really difficult. Okay. All right. The probability of having a normal offspring is 25%. Now, when we talk about X-linked, be very careful. Are we talking about female only, male only, or we talk about the offspring in general? So when you read the question, you have to be very careful. Here it says offspring, the probability of having a normal offspring. So you look at the whole thing, right? 25% normal offspring. Is that 25%? Remember that capital A is the mutant allele. Is mutant. What is a primary school writing? Too bad. So this is normal allele. This is a mutant. So this one affected, normal, normal. Correct? So the offspring, actually the chance is to be normal is 50%. It's not 25%. B, the probability of having an affected offspring is 50%. Right? Again, it doesn't say male or female. So you see, this is female affected, male affected, and if you look at the whole thing, it's actually 2 out of 4, so it's 50%. Most likely this is true. Probability of having a heterozygous offspring is 50%. Heterozygous offspring, there's only one here, right? This is homozygous. How do you call this? You cannot say this is heterozygous. You cannot say this is homozygous. How do you call them? Remember?
Hemizygous.
Hemizygous. Very good. Hemizygous. Okay? So the male are very special. Just like in your class, the males are very special, right? There are how many of you? How many guys in your class?
11, Doctor.
11 out of?
65.
Oh, okay. See, you are special. So males are special. You call them hemizygous. You don't call them this homozygous or heterozygous. 11 out of 65, actually not bad, improving. Okay, now probability for the male offspring to be affected is 25%. Male offspring, remember now here it says male offspring. So you look at the male offspring only way here. So you look at the male offspring, this one is affected, this is normal. So what is the probability for a male offspring to be affected? How many percent?
50%.
80%. So this one is wrong. So the answer is B. For an X-linked recessive genetic disorder, so this is her genotype. She experienced skewed X inactivation, right? Where I gave you a video which explained normal X inactivation and skewed. So she has skewed X inactivation. Now the question asks you, would she express affected phenotype? Yes or no?
Yes, can.
Can. Everybody agree? Is there any possibility that she may not
Yes.
express affected phenotype?
Yes.
Is it that the answer is definitely yes, or it depends?
It,
I think it depends.
it's
Depends on
chance.
what? Depends on what?
It depends on the preferred inactivation of the maternal or the paternal X chromosome.
Exactly. Remember, she is XBA X small. Let's say this capital XBA is the paternal, okay? And then the X small a is the maternal, okay? Let's say, okay, so this one is the paternal and this one is the maternal. So even though during skewed X inactivation, meaning it's not random, it's preferentially inactivating, it could be preferentially inactivating this one, then, okay, so if it is, let me use a different color. If it is preferentially inactivating this one, the paternal, meaning that at the end, most of her cells will carry the X small a, which is the recessive mutant. So she will express the affected phenotype. Correct? So she
Yeah.
will be she will experience this manifesting heterozygote or manifesting carrier. But if on the other hand, if the one that is being preferentially inactivated is this one, so at the end, most of her cells will have the paternal X active, which carries the normal allele. So she'll be okay, right? Because there's no mutant allele, so she will not show the affected phenotype. So when you answer, you have to tell people that although she is heterozygous, but and there is a keyword X inactivation, but she may or may not show affected phenotype because it depends on which X chromosome is being preferentially inactivated. So the key thing is preferentially inactivated. Okay. So she may or may not show affected phenotype. Right? Depends on which X chromosome. So if the one carries the normal allele is preferentially the active X. And if the one carries the normal allele expression activated, then most of the cells will have the X that carries a mutant allele active. Okay. But if the one carries the mutant allele is preferentially activated, then most of her cells will have the X that carries normal allele active. Okay, now you can answer in your own way. This is kind of long-winded, but the key point is you have to tell people that it depends on which one get preferentially inactivated. Okay, no question? Oh, this thing is still here. Okay, Mr. Lim and Mrs. Lim are both carriers of autosomal recessive. Hello, what did I do? So, autosomal recessive. Both of them have blood group AB. Okay, Mr. Lim and Mrs. Lim, both of them are carrier of autosomal recessive. Both of them blood group AB. So now you're asked to use a Punnett square and then state the probability for their first child to be affected by the autosomal recessive genetic disorder and at the same time blood group A. Okay. Now this one is talking about, we are testing about multiple traits. Okay, multiple traits. One is about the genetic disorder. and then the other one about blood group. So from the notes that I've given you, it's actually very simple. How you go about it is I have to clear this first. Okay, so how you should go about it is to actually draw two Punnett square. So you're going to have a Punnett square for Punnett square for the genetic disorder, and then a Punnett square for the blood group. Yeah, I hope you can understand what I'm trying to put here. Okay, so Mr. Lim and Mrs. Lim, carriers of autosomal recessive genetic disorder. It's actually two tables. How do I separate them? How? No border, no border. All right. Autosomal recessive genetic disorder. So the genotype would be, these are both carriers. So it'll be capital A, small a. Where the small a, so Mr. Lin would be capital A, small a. And then Mrs. Lim will also be capital A, small a. Correct? Both are carriers. And then, so you have Mr. Lim. When we do Punnett square, you actually should label them. So this one would be Mr. Lim. And then you have Mrs. Ling here. Sorry, I'm not drawing it here. So she is also carrier. So here you have a small a, big a, a. Okay. So this is the Punnett square for the genetic disorder. Blood group, both of them are blood group AB. So they are blood group AB, so allele A and B. So you have AA, AB, AB, and BB. All right. So state the probability for the first child to be affected by autosomal genetic disorder and descendant have group A. If you look at this one, this is normal, these are carriers, and this is affected. So the probability to be affected is how much? Affected is only 25%. 25% of 1/4. Since we're going to do multiplication, so let's use fraction. Okay, and then from here, blood group A. Blood group A, this is blood group AB, and this is blood group B. So blood group A is going to be group A. So the probability is also. Now, always remember that we're talking about probability. You cannot say that there will be 1/4 A, 1/4 B, and then half AB. Because if they have 4 children or 4 kids, it will not be blood group A and then 2 of them blood group AB and 1 of them blood group B. It will not be like that, because it is supposed to be every single child will have 1/4 chance or 25% chance to have blood group A, 25% chance to have blood group B, and 50% chance to have blood group AB, right? It's talking about probability or chance. So this one is 25%, which is also 1/4. So in order to know that the probability for the kid to be or to be affected by autosomal recessive genetic disorder and at the same time have blood group A, you just multiply the two. So the probability, the answer would be 1/4 times 1/4. How much is it? 1/4 times 1/4.
2
Equals
over
to
6.
1 over 16.
Huh? How many over what?
1 over 16.
1 over 16.
1 over 16. Correct, huh? Wait, huh? Let me check. Huh? This is normal carrier affected. A. If I make mistake somewhere and if you think that something is not right, you have to tell me, because sometimes I can make mistake also. First time doing teaching online. Talking to a screen is very weird, you know. I like to talk to students. Now I don't see any one of you talking to the screen. Anyway, so make sure you know how to do multiplication of fractions. You learn this during primary school. So when you go to university, you should also remember what you had learned in primary school, because almost every time there'll be somebody make mistake here. They'll put 2 over 8 or something like that. So 1 over 16. Okay. No problem. Question 3. What does it mean by no male-to-male transmission in X-linked recessive inheritance? What does that mean? Right? It's actually only telling us that the mutant allele does not transmit from the father to the son. Okay. Just that. Mutant allele doesn't transmit from the father to the son. Understood?
Yes.
Okay. Now, but then if you see this, if you see something like this, this is shaded, huh? Yeah. Okay, and then if we are talking about, say, okay, X-linked recessive genetic disorder, X-linked recessive inheritance. Can you say there is a male-to-male transmission? Wait, forget about this. When you look at this pedigree, can you say there is a male-to-male transmission? Yes or no? Father affected, son affected. Is that a male-to-male transmission? Or maybe I put you a question another way. I'm asking you that, can this be an X? Can this be an X-linked inheritance? We know that X-linked recessive, X-linked recessive. We know that in X-linked recessive inheritance, there is no male-to-male transmission. So now I'm asking you, can this be an X-linked recessive inheritance? You see the father affected and the son affected. Can this be X-linked recessive inheritance or not?
No.
Can or not?
Maybe the mother is carrier.
Exactly, yeah, right. Maybe the mother is a carrier. Therefore, she is XBA, X small a. She's a carrier. She is affected, X recessive So it's X small a y. So this guy, he's affected, of course, he is X small a y. He got the Y chromosome, of course, from the father, but he got this X small a from the mother, not this one. So this is actually not male to male. So this pedigree can be an X-linked recessive inheritance, because this guy, he actually got the mutant allele from the mother. Okay, so don't memorize things like male to male transmission and then you look at this male and male and then say, oh, it's not X-linked. No, don't do that. That's why I say do not memorize mati-mati. So you have to understand it, digest the thing, understand it. Then you will remember it and you know how to apply it. All right. Briefly describe normal X inactivation. I've given you the video. So anybody has any problem with understanding this? X-linked, X inactivation? Anyone got problem or not?
Yes.
Yes, got problem. Okay,
Yes.
Let me put it the other way. Do you understand X inactivation after we watch the video? You
Yes.
understand or not? You understand? Understand means no problem lah. But if you have problem, you have to tell me. I want to make sure that everybody understand X chromosome inactivation, the normal one, and also the skewed one. Okay? Make sure you understand that. If you don't understand, please tell me. So for this one, I leave it now. You go back to the video. But if you cannot understand, you have to tell me, email me, or you ask your class rep to WhatsApp me. We can organize a small session that I can explain to you again. Okay? So go back to the video for this one. Now, what is the main difference between a normal X inactivation and a skewed X inactivation? Remember that in a normal X inactivation, what is the keyword? The most important thing about a normal one is
Random.
random. The X inactivation is random. Random means either paternal, or both paternal and have equal chance to be inactivated. Okay, so equal chance, random. Okay, skewed means it's not random, right? If it is skewed, then it's not random anymore. So for a skewed, meaning that it's not random. So either, oops, so either one can be preferentially inactivated. It's not random anymore. So that's what we mean by skewed. Okay, so that's the major difference between them. All right. Does skewed X inactivation always lead to manifesting carrier or manifesting zygote? The answer is no. Here. Why is it so? And this one we already discussed earlier on, because it depends on whether the chromosome, the X chromosome that carries the mutant allele, or the X chromosome that carries the normal allele is being preferentially inactivated. So you have to see which one get preferentially inactivated. Only then you will know that whether there will be a manifesting carrier or not. All right? Okay, last question. For each of the following, state the probability of all the possible blood groups. This one very easy, and you should be able to do it. Anybody has any problem or not? Let me just give you the answer. If you have a different answer, so this one is A is 50%. Oh, what did I do? A 50%, AB 25 is my percentage, and then B 25. All of you got the same answer?
Yes, doctor.
Yes.
Yes,
Okay, and then this one is AB 50, 50. AB 50, A 50. And then this one is A 50, B 50. Okay, you all got it? Nobody,
Yes.
anybody got different answers or not?
No.
No? All right. Okay, so that is part B. Part C. Of course, in exam, you will not get things like this, but this is just single statement to see how, whether you know them or not. And question can come in any form. Nonsense mutation. So you first must know what nonsense mutation is, okay? Within exons, right? You know what exon introns are, right? Exons are those contain protein coding sequence. Introns are those that do not contain protein coding sequence. So exons are more important than introns when it comes to mutation. So mutation within exon can be dangerous, whereas mutation within introns is not that problematic. So nonsense mutation, what is a nonsense mutation? Nonsense mutation, remember, it's the one that that has a premature premature stop codon. So because of mutation, it creates a stop codon before the actual stop codon. So that's what we call a premature stop codon, right? Just like premature baby born before before term. So this is premature stop codon. So it introduced a stop codon before the actual stop codon because of mutation, insertion, deletion. It can be point mutation. So you get truncated protein, truncated or shorter. Same meaning. Usually non-functional. Okay. So because usually non-functional, so it's deleterious, so it's severe. So nonsense mutation within exon is more severe than missense mutation within exon. Both of them are within exon, but missense, what is missense? Missense mutation, if you remember that it is mutation changing the amino acid. Now, because the amino acid is being changed, so how problematic or how severe or how deleterious it is depends on what is the amino acid that is being changed, depends on which part of the protein. So it's very unpredictable. It can be a small amino acid change to a big amino acid, so it be affect the protein function maybe, but it can be at a part of the protein that is not so important for the function of the protein, then the protein may still be fine. However, a nonsense mutation which create a premature stop codon that usually produce shorter protein. This is usually deleterious to the cell, right? It's usually non-functional. Therefore, the statement say non-some mutation within exon is more severe than missense mutation. So the answer is true. Okay? Missense mutation within exon is more severe than silent mutation. Silent mutation means, what is that? Silent mutation. It's so quiet, silent. Silent means no change in amino acid. It's just like no mutation at all. Whereas missense mutation is changing of the amino acid. So, of course, it's more severe than when there is no mutation. True. Third one, silent mutation changing amino acid sequence. Okay, we already say here there's no change, so this one is false. And then mutation within intron is more severe. No. Remember, introns do not code for proteins, amino acid. Exon is the one that determine the amino acid sequence in the protein. So mutation within intron more severe. This one is false. Chromosomal translocation involves homologous chromosome. True or false? True, false, true, false. Anybody? 50% chance. Guess.
False.
False. Its translocation involves non-homologous chromosome. What is that process that involves homologous chromosome? Exchanging of genetic material between homologous chromosome. You learned about that before? Remember? Meiosis? Remember?
Yes.
Yeah. What is that thing? That cross thing? I forgot already. You learned it in your first year. Remember, they have chromosomes. They form chiasmata. Chiasmata. Does that recall? Yes, no. Year three already forgotten your year one. So that one is exchanging of genetic material between homologous chromosomes. But chromosome translocation involves non-homologous. Nonsense mutation produces truncated protein, right? We already said that, right? Because it's it introduced a premature stop codon. So this one, true. Now, answer the following based on this. This is what we call a notation. A notation. This thing is called a notation. Notation, or sometimes we call it shorthand. What the two words I forgot. So this tells you that it's a chromosome translocation, right? That represents chromosome translocation. True. It tells you that it involves two chromosomes, chromosome 15 and 17. And then it tells you that Q arm of chromosome 15 and Q arm of chromosome 17 is on the Q arm region 2 band 4 of chromosome 15, Q arm region 2 band 1 of chromosome 17. So here it says long arm of chromosome 15. Q is long or short? Long. Q arm is the long arm, so involves long arm of chromosome 15. True. Involves short arm of chromosome 17. False, because it's also Q. If you put P, then it's true. There is loss of genetic material. Usually this one is what we call reciprocal translocation. Reciprocal means I give you some, you give me some. So chromosome 15 gives some to 17, 17 gives some to 15. Exchange. So there shouldn't be loss of genetic material. So this one is false. All right. Any problem? True, true, false, false. Oh, hey, but I got one more question, is it? Okay, how many we got? Section C. Okay. Okay, finish. Oh, good. 3 o'clock. Yeah. Any question from anyone? You have any other question other than this? Maybe when you're going through the lectures, you might, anything that you don't understand, you can bring it up here. You can share your screen, you can just tell. Yes.
I want to ask, is it possible if intron, if injected, can produce functional amino acid so that it will not be able with splicing out?
Can you repeat that?
Is it possible if intron if the intron mutated, is it possible to the intron can produce functional amino acid, which means that the intron will the mutated intron will not be splicing out.
Okay, yes. Thank you for the question. Is there somewhere I can share a whiteboard, right? Okay, never mind that I make use of the white pages here. All right. Yes. So she mentioned, what's her name?
Mira,
Who was
is that?
talking? Mira? Call you Mira.
Yeah, Amira.
Mira. All right. So what Mira mentioned just now, so let's look at this. Let's say this is the exon. This is the intron. This is another exon. So splicing occurs here, right? Spliceosome will X here. Oops. At the junction. Usually if mutation occurs, you know, here, usually no problem. Because this is an intronic sequence, it doesn't code for protein amino acid. But if the mutation occurs very near to the what we call the splice junction, so if it occurs very near to the splice junction, when the spliceosome sits here and trying to do the splicing, it may have problem in recognizing the sequence. The spliceosome, it also interact with the DNA at the exon intron junction in order to do the splicing. So, but because of the splice, the mutation here near the junction, spliceosome may have problem in doing the splicing. So it may actually cause the splicing to be defective. So it can create problems. So this may not be spliced out. This is what she was trying to ask. Yes, it can happen. That's why when we say mutation within the introns, unless if we say it's near the splice junction, then you know that there is a possibility that this will not be spliced up. Then it's going to be problematic. Did I answer your question?
Yes, doctor.
Okay. Any other question?